Muller et al. Where they actually looked back at the results from several previous meta-analyses, and they were all looking at all the studies included in the meta-analyses were studies comparing people diagnosed with depression with people without a diagnosis, called case control studies, of a number of different types of task functional MRI paradigms. Each of the previous meta-analyses, yeah. Oh yeah, my camera. There we go. Okay, cool. Hello. All right. So they look back, each of these 99 studies had said, hey, we found a significant difference between depressed and non-depressed individuals. And each of the previous 3 meta-analyses had said, hey, we find convergent results across these previous individual studies. Muller et al. Is probably the world's best team at doing neuroimaging meta-analyses. They reanalyze the data, fix some of the problems of the previous meta-analyses, and this is what they find. No convergence across those 99 studies, right? So basically 99 studies, each of which claims that there's a difference, put them all together and you get a big nothing burger. That seems like a problem. We can also look at individual studies, right? So this was one that got a big splash back in 2017, which had basically taken people with diagnosed with treatment-resistant depression, done resting fMRI, clustered their resting fMRI results, claimed to find 4 biotypes of depression, and then went on to show that those different biotypes responded differently to transcranial magnetic stimulation. Oh, I could take off my mask, can't I? That's nicer. So really exciting work suggesting that you can finally start to figure out who's going to respond to TMS, right? We know that not everybody does, but some people do. But shortly thereafter, we saw that others were unable to replicate just the existence of these different biotypes, right? And suggests that that you can actually probably get something that looks like those biotypes, even if it's if the data came from a single Gaussian distribution with no underlying clusters. So what do we do, right? We've taken all this money and are not able to provide reproducible solutions to the people who are paying for it. How can we fix this? So, my lab has been thinking about this for a while now. We started a center some number of years ago called the Center for Reproducible Neuroscience that aims to basically, come up with ways to try to address these issues. One is around open data. I'm not going to say much about data sharing today. You'll hear more about that later, I think, from Satra. We've developed a couple platforms, one called Open Neuro, which shares complete raw neuroimaging data sets, one called NeuroVault, which shares sort of summary statistics. We've also been involved in the development of open tools, particularly focused around the brain imaging data structure, which I'll say a bit about later, things like MRI-QC and fMRI prep. We've been deeply involved in the development of a standard because we realize, as I'll say more later, that without having a common way to organize your data, it's impossible to effectively share data. And I'll also say a bit later about some of the kind of meta-scientific work that we've been doing to try to understand how well is our science actually working. So I like to think of this as a design problem. Does anybody know what this thing is? Any guesses? It's a commercial grade paper cutter. So the idea is like you stick a whole ream of paper in right here and it like chops it off. You see there's 2 green buttons on this machine. Why do you think the machine has 2 green buttons? Anybody have a guess? So basically people don't cut their hands off, right, by holding, because the obvious thing you want to do is hold the paper in and hit the button, right? This prevents you from doing that. So this is a design that explicitly anticipates the stupid things that people will do and prevents them from doing it. And that's one of the things that design is, especially industrial design, is all about, right? Figuring out how do you want people to behave and how do you want them to not behave, and pushing them to do the things you want them to do and making it harder to do the things you don't want them to do. Now in science, one of the big challenges is that these are like a lot of the things that we want to incentivize or not are cognitive things, right? And it turns out that humans, as pointed out in this article a while ago in Nature, are really good at fooling ourselves, right? At making ourselves think we see something, we see a face in the clouds, we see what seems like non-random behavior in random phenomenon, all these sorts of things. We aren't very good at thinking sometimes. And we think that is a major part of where a lot of these problems in science have come from. So there's an idea from behavioral economics that I like to think about called choice architecture. And the idea is like whenever we go into a situation, be it walking into the store to figure out what to buy or sitting down with a software package to figure out how to analyze data, there's features of that environment that drive us towards certain choices and away from certain choices. So for example, if you're using SPM to analyze your fMRI data and it has 0.001 as your default threshold for cutoff, for thresholding, you're very likely to just use that default, right? If it was a different default, you'd be very likely to use that different default, not because one you think is better than the other, just because that's the default. So Thaler and Sunstein developed this idea of nudges, which is a somewhat controversial idea still about whether it actually works. But nonetheless, I think it's a compelling idea for thinking about how do we want to try to fix science. And the idea is, one, we want to incentivize the right behaviors. We want to use the power of defaults, make sure that the defaults are the right defaults, give feedback to people at the right times, and in particular, sort of think about what errors we expect and try to help people avoid those errors. Okay, so what are some of the things that we need to think about then? How many of you, show of hands, how many of you have read this paper? Okay, this needs to be on the required reading list. Ioannidis 2005, Why Most Published Research Results Are False, which was a really big splashy paper back in 2005. And I think, even though there's still controversy about some of the specific claims that he made, I think the general claim most people now would agree with. So in this paper, I'll come back in a bit to the statistical argument that he actually made for that claim in the title. But he also in the paper laid out a set of features of science that he thought sort of would make a particular subfield have more or less reliable science. So for example, the smaller the studies, the less likely the findings are to be false. The greater the number and lesser selection of tested relationships, that's how many things you're looking at, the less likely they are to be false. The greater the flexibility in designs, definitions, outcomes, and analytical modes, the less likely they are to be false. So that each of these should already be ringing a bell to you if you've ever touched neuroimaging data. So I'm going to walk through each of these and sort of talk a bit about what we think some of the issues are. And the first, it has to do with statistical power, right? So this is, I should highlight that this is really focused on studies that are, trying to either do kind of null hypothesis testing, to say, where is there activation across the brain, or studies trying to do sort of some kind of machine learning predictive analysis. But this is particularly focused on null hypothesis testing because that's where the idea of statistical power comes in. So power is basically the likelihood that if there is a true effect of the particular size that you want to find, your study will find that effect, right? So this paper by Button et al. Was the first to really sort of make the argument about very low statistical power in neuroscience, looking both at human neuroimaging and rodent studies. This is a more general study by Munifo and colleagues, which looked across lots of different domains of biomedical science, showing that the same pattern exists. So what do you see here? You see that, on the order of a third of all studies have, power of 10% or less. That means that, even if the effect is there, 90% of the time, they're not going to find it. If you were trying to run a clinical trial that was a medical clinical trial that had 10% power, they wouldn't allow you to run it. You would be told that study is futile, right? Because there's hardly any chance of actually finding the thing, even if it's there. Now, this is the argument that Ioannidis made in his paper, and it's sort of laid out nicely in the Button paper, which is, you might say, well, I'm controlling my type 1 error, my false positive rate, right? So I'm going to, do 100 studies, and even if there's nothing there, I'm only going to find 5 false positives if I have 5% false positive rate. But Ioannis pointed out that there's something else that we might care about, which is what we call the positive predictive value. So not just looking at all findings, just looking at the positive findings. So let's say you're a physicist and you're trying to measure something. Unbeknownst to you, your measurement device is broken and it's just giving you white noise. And so you go do 100 experiments and you get five positives, all of which are false positives. So your false positive rate is 5%. But all of those positives are false, right? So your positive predictive value is 0. None of the positive results are actually true. And once you fix the machine, then the more powerful the machine is, the more sensitive the machine is, and the bigger the effect is, the more likely you're going to be when you find something positive for it to actually be a true positive. The other thing that actually Ioannis didn't point out, but others have pointed out, is that when your power is low, you're only powered to find very big things. So let's say your power is very low and the only correlations you can find are, say, 0.8 and up. So that's, the literature is going to be full of reports of very strong correlations, even if the actual correlation is only, say, 0.2, because those are the only ones you can find. And so this is what people call the winner's curse, the idea that low power results in substantial overestimation of effect sizes. I'm going to show you a few examples today, and I'll dig up a link to the notebooks where this comes from. I'm going to show you some examples of just using real data to try to look at this. And I'll use data from this thing called NHANES, which is a large data set. It's available in R, and I have a Python package if you want to use it in Python. And it's nice because it's big enough for us to sort of treat it as an entire population. So I wanted to look at the correlation between BMI and this thing called HbA1c, which is the measure of blood glucose. And each of these little faint lines is basically one sample that grows over time, right? So I first take a sample of 10 and then 20 and so on out to 500. And what you see is that when the sample sizes are small, right, the true correlation is here across the full sample, the true correlation is like 0.2, right? And even when the sample size is 500, you can get values anywhere between, plus or minus 10%. But when the sample size is really small, when it's say 50, you can get anything from like a 0.8 correlation down to like a minus 0.2 something, right? So the idea is that correlations take very high sample sizes to stabilize. Many of you are probably familiar with this paper that just came out in the last year by Marek et al., which it basically made exactly this point with neuroimaging data. This is using I can't remember if this one, I think this one was from ABCD. They also looked at Human Connectome Project data, looking at both structural and resting state functional measures, and basically showing that with small samples, you can get a huge range of correlations. And this is probably the most striking thing with sample sizes of 25, you can find one sample that has a positive correlation of 0.5, another that has a negative correlation, minus 0.64, even when the true correlation across the entire sample is minus 0.08, right? That's just because of the noisiness of those small samples. Especially with correlations are particularly sensitive to this. When you combine that with publication bias, you can see entire literatures that can go astray. So around 2,000 people gained the ability to measure relatively cheaply and easily genetic variants in individuals, right? So, and so you started seeing a bunch of studies that would look at a single genetic variant. We call these candidate gene studies to see if those variants were related to either a behavioral finding or an imaging finding. This is a meta-analysis of one particular of those. This was a genetic variant in this gene called the brain-derived neurotrophic factor gene, which was thought to relate to hippocampal volume. The size, so this shows year of publication, reported effect size, and the size of the dots is related to the sample size. I don't have the legend on here. But you can see that early on, they were all positive findings, and they were all fairly small samples. This one was not very positive, but these were all very large effects. The largest effects are always the smallest samples. Over time, the sample sizes get bigger, the effect sizes get smaller. This is what's become generally known as the decline effect. And we now actually know that none of those, the literature around 2000 and 2010 was full of these candidate gene studies. none of the candidate genes that were investigated with regard to hippocampal volume or other brain phenotypes, for the most part, have turned out to actually replicate. Now that we have genome-wide studies with tens of thousands of subjects in them, we're able to find other replicable findings, but none of those candidates replicated, even though there were many different studies and there were meta-analyses showing that they replicated, and that was all because of publication bias, we think. So how well powered are our fMRI studies? This is, we published a paper in 2017 that did an initial version of this. This is an updated version with the data through 2020, primarily from Neuroscience, which you'll probably hear more about at some point. Basically, we just use the text to pull out the sample sizes. So there's some noise here. But what you can see is that sample sizes have gone up over time. This is a log scale, so it's actually going up more impressively than it looks. So, there's now in the last decade, there's a lot of studies with sample sizes over 1,000 where there were really none prior to, whatever that guy's about 2009. So using some assumptions, you can estimate what is the size of the effect that this study was powered to find. And the idea is, like, a study should be powered to find a reasonable effect size scientifically, right? So if I'm doing a study looking at height between individuals, and I'm only powered to find a difference in height between 2-year-olds and 18-year-olds, right? That's probably not an interesting effect size, right? So we need to figure out what's an interesting effect size. The median study was powered to find a single voxel with a Cohen's d of 0.73. That's a 0.73 standard deviation difference between the groups. The question is that plausible? So we can't look at the reported effect sizes in papers because those are done after thresholding, so they're always going to be inflated. So what we needed to do was to estimate kind of what are realistic effect sizes for various types of tasks. So we used the Human Connectome Project data to do that. And fortunately, they had given us a set of regions of interest. So we took those regions of interest and then we sort of overlapped them with Neuroscience. So for example, if we're looking at a motor task, we would take the regions that the Human Connectome Project had given us that were regions of interest, and then we said, give us only the voxels within those areas that also show up in Neuroscience. The idea is we want to be really sure that these are voxels that should be related to motor function. And then we compute the effect size based on the unbiased effect size based on the Human Connectome Project data within those areas. And importantly, these regions of interest were written down before the HCP data were ever collected. So they're a priori regions of interest. And this is what you see. So on the left, what you see is the percent signal change in the bold signal, right? That's how much does the signal go up on average within a person. And in general, the biggest that we ever expect is about 4%. And that would be in the kind of primary, visual or somatosensory motor cortices. And you see that we get there, like for the motor task, we're up around, this is 4%, or around 3-4%. A bunch of these other tasks are, above 2%. So those are whopping effects for fMRI. These are like really gross, it's like move your hand versus do nothing, right? That's going to give you a huge increase in brain activity. So they're already kind of, probably the biggest effects we would ever expect. Now, the problem is that when we look across a group, we're not looking at how much do people's effects go up on, or how much does activity go up on average. We're looking at how much does it go up on average with respect to the variability across people. Right? When we compute a t-test, the thing in the bottom of that t-test is the variability across people. And so we can quantify that using that Cohen's d measure. And here we see that the biggest effects are ones that are like a Cohen's d of about 0.8. That's just starting to reach what we would think of as a large Cohen's d. And most of them are in the kind of the medium range. So what this is saying is that the most whopping effects we'd ever expect in fMRI are barely at the point where the median study is powered to find them. So half of the studies that were published in 2020, I think that was the year, weren't even well enough powered to find the biggest effect that you would hope to find with fMRI. This is, I'm colorblind. I don't know why I color coded things because it's very hard for me to see the differences here. And usually I would gripe about somebody doing this. I've color coded here by studies that have less than 20 participants in one of the groups. These are all the studies from that Muller meta-analysis. And I'm calling less than 20 grossly insufficient for a case control study, right? If you're comparing two groups, there are studies where N of 20 is perfectly fine. We can talk about those later. But for a case control study, if you're comparing two groups, I would argue, and I'm not, actually, the argument doesn't come from me, it comes from this Simmons et al. Paper. This is this paper that was titled False Positive Psychology, where basically they made a bunch of recommendations about how to improve the reliability of psychological science. And one of them was authors must collect at least 20 observations per cell or else provide a compelling cost of data collection justification. And nearly all of these studies had, the majority of them certainly had insufficient Ns. So that means that if you just take all those studies, there were 634 individuals across those studies who volunteered for fMRI studies that were almost certain to guarantee either null or false positive results because they were just so badly powered and generated about 224 tons of CO2 if you make some assumptions about how much CO2 is generated by an MRI scanner. Okay, so will machine learning save us? Gael Varacqua has a great paper from a few years ago titled Cross-Validation Failure, where he started out looking at just some simulations and also just kind of theory about the variability in predictive accuracy in relation to sample sizes. And he shows in both simulations and just using a binomial that the larger the sample size, the lower the variability in predictive results, just sampling variability across samples. And I looked at this in that NHANES data set again, just to show you that you just take any real data set and it's going to work. This is building a predictive model to try to predict whether someone is a smoker or not based on their age, their gender, and the amount of cadmium in their blood, which turns out to be a reasonable marker for whether you're a smoker or not. And the true predictive accuracy on the full sample is around 70%. But you can see that with a sample size of 100, you can get anything from 0.4, which is less than chance, up to perfect accuracy. Just highlighting the variability. And he saw exactly this when he looked at the literature. Well, not exactly that, because the literature doesn't show the bottom of that, right? These things down here don't make it into the papers. These up here make it into the papers because of publication bias. So that's what you see when you look at the literature for almost all these different domains. As sample sizes go up, reported predictive accuracies go down, right? That's not because it's actually going down with bigger samples. It's because somehow we know the smaller samples give you a better chance to find higher accuracies. And there's some kind of bias or questionable research practice that's filtering out the bottom half. Okay, so the problem here is, right, you want to get a PhD, right? And you can't, let's say that you care about, like, what are the biotypes of depression? You probably don't have the money and time in your PhD to go out and collect thousands of individuals to be in your study. And this is, when I used to go give talks at places and have the lunch with the trainees, we'd always have these discussions about, what do I do? So I wrote a paper a couple of years ago called The Cost of Reproducibility that tried to grapple with this particular challenge. And I don't think I have great answers, but I have a few suggestions. One is like this quote from Nancy Canwisher: if you can't answer the question you love the question you can. Right. So you may have to pivot, right, and realize that you might not be able to ask the ultimate question until later in your career when you can go get the big grants to do the big studies. Or you might find other questions that are more proximal that you can actually find interesting. So how has this worked across different fields? One is, collaborate. If you look at genetic studies, nobody's out there collecting 100,000 individuals and putting them into a genome-wide analysis. You look at the author list of those papers, they often have 300 authors on them, right? Because lots of people are contributing data to each of these. They've developed these consortia, and I think that's been really effective. And it started happening in neuroimaging with the Enigma consortium. So there are ways to work with others to get your hands on data. There's increasing amounts of shared data every day across all the different archives that are out there. And another thing to do is like spend more time focusing on theory and computational work, and with the goal of ultimately applying that. I just want to tell you about one success story. This is Mac, who was a postdoc in my lab for a couple of years. He's now at University of Sydney, and he published. I don't want to like fetishize publishing in fancy journals, but he published in a number of fancy journals, right? And did it all based on shared data. He collected none of his own data the whole time he was in the lab. So it just highlights the fact that it is possible to be a what some people call a data parasite, and to build a successful career, asking really interesting questions. And what it comes down to is, and the reason that he's been able to be successful is he's come up with interesting new questions that drove him to interesting new ways to look at data. OpenNeuro is the platform that we run for sharing of raw neuroimaging data sets. Now includes MRI, PET, MEG, EEG, intracranial EEG, soon to include fNIRS as well. There's now 720 data sets, more than 27,000 participants, mostly human, but some non-human data as well. And so that's even if you're ultimately going to be collecting data, it's a good thing to go see what data are out there to at least kind of benchmark your methods, your analysis methods before you start analyzing your real data. Any questions? Sorry, I cannot hear you at all. I have poor hearing and masks and noisemaker. I'm going to come back here. Do you want to write it in Slack, or do you want to say it to me? Okay, good. Very good questions. I'll repeat both of them. Thank you. So the first is like, if we're all using shared data, who's going to collect the data? And clearly people are going to, I think, keep collecting data. And we just have to figure out how to sort of reuse them. But one thing to note is that increasingly there are these large consortia and sort of publicly funded projects like the Human Connectome Project, like the ABCD project, that are collecting very large data sets. Unfortunately, they often end up collecting tasks that are pretty boring, right? Because you're going to be putting this much money into it. You don't want to use like a risky task. And so there's always challenges with that, right? But I think it's there are enough people collecting data now, and it's just a question of, can you find the data that are relevant to your hypothesis? The second question had to do with kind of like the idiosyncrasies of all the, like, you go look online, and let's say you're interested in reading, and you go find the 8 studies on OpenNeuro that are doing fMRI of people doing reading tasks, but they're all going to be like different scanners, different populations, different tasks. And sometimes you can, kind of harmonize those things, and sometimes you can't. But often, in general, the scale of the data can sometimes allow you to kind of like, generalize across all of those idiosyncrasies. Now, sometimes it really depends. You have to understand the idiosyncrasies to know, like, are these ones that really matter? Are they going to really change the interpretation? Or are they sort of things where we would hope that our results would generalize across them? And that's a difficult scientific question. Yeah. Yeah.
Studies in people with lesions or brain damage or interventions and EEG and other modalities. Yeah. So would you say that using other modalities and lesion studies can help us understand what's going on with fMRI?
Yeah. So the question is, obviously, fMRI is not the only method we have, right? People do electrophysiology studies like EEG, MEG, they do lesion studies. And the question is, like, should we, if you're an fMRI researcher, should you be thinking about other modalities? And ultimately, it's a scientific question, one of like, are there for example, lesion studies that are relevant to the particular thing you're studying. In general, I think that, lesion studies have historically been really important, and they remain really important for understanding whether some particular brain system is necessary, right? Just because something is seen to be active in an fMRI study doesn't mean that if you remove that thing, the person will fail at the task, right? It could be something that's like monitoring or along for the ride. And so I think that lesion studies remain really important. Now, the problem is that lesion studies are often very small. And that's when you have to think about moving away from this, like, large sample, small data per individual regime to a much denser regime where you're studying individuals much more intensely, right? And maybe give up on like generalizing across an entire population. But if you look at the way that people in visual psychophysics and visual neuroscience have historically worked, right? Often those papers will have 5 subjects, 3 of which are the authors on the paper, right? And in part that's because, like, well, you would hope that like the contrast response to area V1 doesn't change a lot across people. It doesn't seem to change that much across species, right? There are things that probably change a lot more across individuals, and those are issues where that strategy might not be as effective. Yeah, so I think that it really comes down to what's the scientific question you want to ask, and then how can you find a feasible, reliable strategy to get answers to that question. Yeah, sorry, I'm going to come over there because this thing is noisy. Yeah.
You have an idea on the prospect of like what studies from the 2000s actually stand the test of time, and also like has your mindset been like, oh, like none of that is actually valid, and now we need to actually like see what is actually valid, or because it could be so easy, like doom and gloom.
Yeah, that's a really good question. So the question is basically like, looking at what I'm telling you, you might think that any fMRI study published in the 1990s is just completely unreliable, right? And that is not the case, right? One of my most highly cited papers is a paper back from like one of my first imaging studies looking at different aspects of language processing in the prefrontal cortex. I think the study, it was a combination of like a meta-analysis and an fMRI study. I think it had 8 subjects in it. We aligned, this is before you had automated alignment. So we would like align each slice by hand to the atlas because we only had a few slices of the prefrontal cortex. And that result has turned out to be really robust, and it's now been replicated in much bigger studies. So it isn't the case that everything from back then was bad. I think that my intuition is that the kinds of results that are at most risk, given everything I'm telling you today, are brain behavior correlation results, because we know that you need much bigger samples to find brain behavior correlations, and group differences, right? Because that's similar to a correlation, right? Looking at differences between groups. I think that, in general, I mean, we know there are a lot of things that are super reliable, right? You put pretty much any healthy adult into an MRI scanner and show them faces, you're going to find a face area, right? There are things that basically show up for everybody. It's these kind of next level sorts of questions, especially when you start thinking about distributions, and you're just not, if you look at those plots I showed you about, how sampling variability changes as a function of sample size, you didn't need to actually do anything to find those. Those are all just theoretically evident from the central limit theorem. But it took our field a long time to kind of come to grips with those findings. Okay, so what I want to do now is talk about analytic flexibility. This is from that 2017 Nature Reviews Neuroscience paper. We wanted to know how many plausible workflows could one find for our fMRI data. This is only using FSL and taking just at each step, some of the kind of reasonable things one might do at each step, putting them all together, and you get almost 70,000 different workflows. Right? And obviously, there's probably at least this many for any other software package, and pretty much an infinity if you're writing your own code. And we know from Josh Karp's early work that these are actually used in practice. There's high variability. He looked across 241 papers and found that there were nearly as many unique analysis pipelines as there were papers. And people were all over the place in terms of the package they used, the smoothing they applied. This 8 here is probably because that's the default in SPM, right? Or at least it was. Different inference measures. Josh also did a study looking at whether this has an effect. He took a data set from what was then OpenFMRI and now OpenNeuro, and he analyzed it through almost 7,000 different pipelines, and looked at the range of the statistics at every voxel in the brain to ask how much variability is there in the outcomes. And so on the left is the average activation across those pipelines, which fortunately looks, this was one of our data sets. This looks roughly like what we had reported. But if you look at the range, this is like, up to 7 or 8 standard deviations from like the pipeline, the statistic in the pipeline for the highest result to the statistic in the pipeline with the lowest result, right? So huge differences in the potential result there due to differences in analysis methods. And this is another place where machine learning can make it much worse, right? This is a fun paper from Skosik et al., where they basically show that you can very easily, by just sort of iterating, like using machine learning to find a particular pipeline that can basically give you signal out of noise, right? So there's no actual signal here, but they find, supposed classification accuracy of, 0.6 something, simply by modifying hyperparameters in what seemed like innocuous ways. I don't want you to think that this is all just fMRI problems, right? Steve Luck and Nicholas Gasplin had a great paper basically showing that, many of the same things that are done in the ERP literature can result in very high, what they call bogus effect rates. So we think that pre-registration is a really important way to battle this. Obviously, it's not a cure-all, but we've started trying to pre-register all the analyses that we do. And the idea is you go to a place like Open Science Framework and you basically write down what it is that you're going to do. What's your sample size going to be? What are your inclusion, exclusion criteria going to be? How are you going to analyze the data? What's the hypothesis you want to test? We like to do it, even though we've not done this in most cases, but we'd like to do it actually with code that's already running based on simulated data. And I'll talk more about that later. Now, I used to worry that, the focus on pre-registration would prevent people from doing exploratory analysis. And, the idea is if the only analyses we ever do are the ones that we thought of before we see the data, then we're probably going to miss out on lots of interesting stuff. The question is, how can we remain able to find that interesting stuff, yet not sort of fool ourselves into thinking we found something when we haven't? Part of what pre-registration does is it just makes it really easy for you to remember which of the things you did that were planned beforehand, which of them were exploratory based on the data. This is what happened in the medical clinical trial world when pre-registration came to be required. So in 2000, the National Heart, Lung, and Blood Institute in the US started requiring anybody doing a clinical trial to register their primary outcome. That is, what is the one thing you want to measure? Is it like reduced deaths? Is it, reduced disease? What's the thing you want to measure? Because you can often measure a bunch of different things. This is showing the relative risk of some outcome, like, death or disease. And if you're below with the line here, that means that your treatment worked. And if it's a solid, if it's a plus, that means that the authors reported a positive effect. 2000 comes along, drugs don't work anymore, right? Most of them are now sitting here right around 1 with null effects, a couple of positive ones, and then 1 actually reported harmful. It's not as if drugs stopped working in the year 2000, right? What happened was people lost the flexibility to make it look like the drugs worked. And so we now know that, there's a lot of people probably walking around taking, this drug that may or may not be actually having an effect. Now, pre-registration will prevent p-hacking. It'll prevent a number of sort of questionable research practices, but it doesn't get rid of analytic variability, right? You can pre-register one analysis plan, I can pre-register another. And so we wanted to look at how variable analytic methods actually are in the wild and what sort of effect this has. So we did a study really led by Rotem, who I think is a NeuroHackademy alumnus, and Tom Schoenberg in Tel Aviv, working with a bunch of others to basically ask what happens when you collect a data set and give it to a bunch of people and then look at the results. So Tom and Rotem collected a data set using a task that we had published previously. As a gambling task, I show you two numbers and say, would you take a gain and a loss? Would you take this gamble? And there were two slightly different versions of the task. And so we collected data on 108 people. We asked people through social media and at conferences, would you analyze these data? We'll give you all the data. You can have as many as 3 people on your team. You'll get about 3 months. The one thing is you can't tell anybody outside of your team what you found until we lift the embargo, after everybody has submitted their results, because we didn't want teams talking. So they all signed a non-disclosure agreement, and we offered them co-authorship on the paper, which is why the paper had about 200 authors on it. We gave out the data set to 82 teams, ended up getting back from 70. And we gave them the time to answer 9 questions, which were all of the form, is there activation in some particular region for some particular feature of the task? For example, does activity in the ventromedial prefrontal cortex go up as the amount you could win on the gamble goes up? And we told them to analyze the data as if they were analyzing a study from their own lab. Just use your own workflow. The only restriction was they had to do a whole brain analysis. So we got the results back from the 70 teams. We also got a bunch of additional results back, like thresholded and unthresholded statistical maps, in addition to their yes/no answers, so we can try to figure out sort of what was going on. And we asked them to give us a detailed description of the of their workflow. And based on this report that we published a few years ago called the COBIDAS report, that basically lays out kind of a checklist for, these are all the things you need to describe about your analysis workflow. None of the 2 teams no 2 teams out of the 70 used an identical workflow. And what was even more striking, like many of them were giving us 15-page long descriptions of their workflow. We still couldn't tell often exactly what they had done without actually getting the code or sort of intermediate files from them. So here's the main finding. The y-axis here is the proportion of teams saying, yes, there is a positive result in this area. And the x-axis is the 9 different hypotheses that are sorted by that y-axis value. So if everybody agreed, these numbers would all be either 0 or 1, right? Either everybody would say there's no effect, so it would be 0, or everybody would say there is an effect, so it would be 1. And we do see, for 3 of them, these are all like between group comparisons, between subject comparisons in the amygdala, I think. Those basically hardly anybody finds anything. For one of the hypotheses, pretty much everybody finds this is effect of losses in the ventromedial prefrontal cortex. Pretty much everybody finds that. For these other 5, basically 20 to 40% of the teams find an effect. So another way to look at this is across teams, there were, of the 70 teams, there were 33 different patterns of yes/no answers. And for any particular hypothesis, there are at least 4 workflows that could find a positive result for that hypothesis. We can also look at the whole brain results and ask, at any particular voxel in the brain, what's the proportion of teams saying yes, where this passed their thresholding? This is like the strongest result of all the 9. This is for the one that was that everybody agreed on pretty much. The voxels here in the middle of the ventromedial prefrontal cortex, or a few other places, the voxel with the highest overlap between teams only had about 3/4 of teams saying, yes, there's activity in that voxel. So not very good agreement even at that level. Another way to look at it is, like, for any particular hypothesis, what's the minimum number of voxels that were reported across the brain? And for each of them, the answer was 0. What's the maximum number of voxels for a particular team, right? And here it's often 135, 120,000, 147,000, right? So you can go from one team saying there are no active voxels to another team saying half the brain is active. Even for these ones where I forget what the numbers were, where pretty much everybody agreed there was nothing there. So we also looked at the underlying the whole brain unthresholded statistical maps and asked, how do they relate to one another? So this is a correlation matrix. Each row and column here is one of the teams. And what should really pop out at you is, there's one big block of teams up here that are relatively highly correlated with one another. But even the green and red up here are basically, did they say yes or no to this hypothesis? This is one that had, I think, like 30-something% agreement. And you can see that even teams that were highly correlated with one another often gave different answers for the yes/no question. But what's even more striking is that there's a set of teams down here whose results were anti-correlated with the other teams. So this is what the average map looks like for this team, this set of teams up here, right? We see our ventromedial prefrontal cortex activity. This is what we see for this other set of teams. This looks like a standard kind of task activation map, right? With deactivation in the ventromedial prefrontal cortex because it's part of that default mode network. And when we dug in, we realized that basically we could find all of these teams that we could get enough details on mismodeled the task. And so their model was picking up on overall activation rather than the more specific parametric effect that we were looking for them to find. And then there's a set of kind of noise teams out here that were really uncorrelated with anyone else. Nonetheless, if we put everything together, we can find even including those ones that did it wrong, we can find converging results using a meta-analysis at the level of like all the different analysts. And this is what we've come to call multiverse analysis. I mean, people in the statistics and psychology literature have been talking about this for a while. So there is stuff in there, right, which we can find at the level of this meta-analysis. And so we've started thinking that we really want to try to start using multiverse analysis to improve the generalizability of our findings, right? And also to understand, like, what are the analytic knobs that cause us to find something or not find it? And so we've been doing a lot of work to try to build software tools. You know, the problem is you don't want to go in and run 100 analyses by hand and then have to try to put them all together. So Chris Markovich in my group has been developing this thing called FitLens, which basically takes a description of a statistical model, which is written using this thing called the BID statistical model specification, and then basically applies that to a data set and gives you back results. And it'll allow us to sort of automate multiverse analysis. It's kind of like, it's still pretty bleeding edge right now, so it's not really ready for prime time, but we're hoping that within the next year it will be. Okay, time for a poll. How many of you have written computer code in the course of your research? How many of you have ever been trained in software engineering? How many of you have ever written a test for your code? Oh, wow, good. So we think that, you know, everybody is writing code in science these days, pretty much. And we think that there's a likelihood that a lot of scientific results are probably potentially tarnished by software errors. This is kind of the best known example of this. Jeffrey Chang's a structural biologist who published a bunch of papers in the early 2000s about this particular protein, the structure of this particular protein, the ABC transporter, all of which had to be retracted because a particular piece of software that they were using, and my understanding is actually it's a piece of software they'd gotten from another lab, had one sign change in it and basically totally changed the inferred structure of the protein. Had to retract a total of 5 papers. So I've been writing for a long time about software errors because we've seen them in our lab, and you know, this is a big driver for why I've been, you know, giving talks like this for the last few years. So I was doing this is an analysis I was doing way back, almost a decade ago, just doing classification on a bunch of data sets from open fMRI. And so I write my little cross-validation thing here, and I get 93% accuracy at decoding what task the person was doing based on their brain activity data. If it was 99% or 100%, I would have said, hey, something's really wrong here. But 93% is like, wow, that's really good. It's probably too good, but it might be believable. Right? So I sent the code to someone who was then a grad student who then became a postdoc with me, Sandy Koyejo, and he looked it over. He said, oh, dude, you left out the colon here. And when you do it right, it's still way above chance, but it's far from 93%. Fortunately, this all happened before we had ever submitted the paper. But it just goes to show you how something very tiny, you know, the code doesn't crash, right? It just does something unintended. And we saw this more recently with a preprint that we had posted, where we had done analyses of the ABCD data set, looking at their implementation of the stop signal task. This is just behavioral analyses. And we found that, a decent proportion of the subjects exhibited this particular phenomenon that we thought is really problematic for the assumptions that go into the model for the stop signal task. We posted the preprint and we made all the code available, and we were beaten pretty hard on the ABCD team, so they obviously dug into our code, and Sage Han from that team found an error in a really kind of hairy bit of Boolean logic here. And if I remember correctly, I think one parenthesis was in the wrong place or something like that, so it was grouping the data incorrectly, and the actual result was not 1,300, not 18%, but about 6%. Still, it didn't undercut the main argument of the paper, but it's a substantial difference in the number that we didn't see. And in part, I think this reflects this phenomenon that I've come to call bug hacking, which is if that code had given us 0%, we would have dug into the code to figure out what was going on, because we knew that some of them had to give us, had to be showing that violation. If it had given us 50%, we probably would have said, hey, wow, that's amazing evidence for our claim, right? So the idea is that when bugs that are, countervailing to our hypotheses, or that give us bad results, are much more likely to be dug into than bugs that work in our favor. So we've started working according to what I like to call the principle of assumed error, which basically means that if you find something that looks really good, start by trying to figure out what you did wrong to get it. And this is at least one way to try to immunize yourself against bug hacking. I'll come back in a bit and talk about other ways. So, coding is not something anybody's born knowing how to do, right? It's something we all have to learn to do, and it's kind of like a craft. It's like, learning how to build furniture or something, or how to fix a car, right? In the sense that it takes time and it takes practice and it takes effortful learning. And so we've become really sort of, interested in thinking about how to improve the quality of coding in science, because as I'll talk about in a few minutes, I think that coding quality is really essential to help reduce the prevalence of errors. So what do we mean by high quality code? Well, it should be, more than anything else, it should be readable. And I'll say more about that. It should be robust in the sense that it shouldn't do the wrong thing if it gets bad input. It should fail in ways that are informative. It should be modular in the sense that it can be sort of tested in parts and reused in parts, and it should be well tested. And as I said, as the analogy of software carpentry, if you're not familiar with software carpentry, it's a really great group with a ton of good resources for improving coding skills. The only way to get better at it is through practice. So one of the kind of saws in the software engineering literature is code is read much more often than it's written, right? And often the reader is you down the road. And you have to figure out what on earth was I doing when I wrote this code a year ago? Many of you probably had that experience of trying to figure out what you were trying to do, right? And so we've come to think that writing well-styled code, readable and well-styled code, is really important. Now there are both language independent principles of software design, and sort of language-specific conventions. So if you're writing in Python, and I'll talk a bit about some of the tools you can use, because I'm going to focus on Python. There are a lot of tools to help you do this. These are some of the books that I would recommend. If I was going to start with one, I would start with The Art of Readable Code. It's relatively short. You can find a PDF online. And it's language independent. It really lays out how should you think about writing code that will be readable by you and others in the future. Regularly, you see people on Twitter write, "Oh, I need to go back and comment my code before I put it up on GitHub." And I think there's a general thought amongst a lot of people that the difference between good code and bad code is how many comments are in the code. And this is, if you read the software engineering literature, nearly everybody sort of disavows this notion, right? The idea is that, sometimes you need comments. You need comments to help the reader of the code understand the things that can't be made obvious from the code itself. But in general, the reader should be able to read the code, and from the naming of variables and the structure of the code, should be able to basically figure out what the code is doing. So don't comment bad code, rewrite it. We've started doing code review in my lab, and I think it's been really useful for us, both as not so much as a way to find errors in code, but to really sort of help people, help the people in the lab who aren't great coders level up, because they see, what goes into thinking about how to code. But particularly, having someone, what we do is mostly like, I or someone else will read the code that one of the people in the lab submits for review. And it's mostly just trying to figure out can I tell what this code is doing without having to ask that person any questions? Can I tell why they did something? What are they trying to achieve? Why did they do it? And then sometimes we'll see, oh, there's a much cleaner, better way to do this. But often it's just about trying to figure out the readability of the code. And this paper by Miriam Petrie and Greg Wilson lays out a lot of ways to think about how to do code review in a group. We've also become much more sort of interested in the use of testing. And so there's a lot of different types of tests you can do. And unfortunately, most of what's in the software engineering literature is not super useful for the kind of stuff that we are going to do as scientists. Some of the types of things that we think are important are, one, just what we call a smoke test, which is like, will everything run? And especially this is important when you're making a bunch of changes, and you just want to make sure that you haven't done anything that's broken the whole machine. Another is sanity checks. There are certain things that should be within particular ranges. Make sure that they're within those ranges. I'll show you an example of that in a second. And then, when you're writing functions that are going to be reused a bunch, making sure that you have unit tests to actually exercise those functions and make sure that they give you the right answer when they should, they give you the right error when they should, they fail when they should. We think that's also really useful. This is a paper that was published in PLOS One back, I think, 2015, that had made some arguments about relationships between conspiracy theory and rejection of science. And they had, in their analyses, had regressed out a bunch of sort of potential confounds, one of them being age. PLOS One requires sharing of all the data. Somebody looked into their data and said, hey, wait, you have your data, your subjects are supposed to be adults. You have one who reports an age of 5, one reports an age of 32,757. That's not a human, right? It's not a human age. And so they had to go back and publish a correction, basically saying, hey, when we take those out, the results actually change a little bit. That would have been easily fixed by just having an assertion saying, hey, let's just make sure that all the ages in our data set are actually within the range that ages in our data set are supposed to be. There's now an increasing number of tools that you can use to basically automate this type of testing. GitHub Actions is probably the one that's become most popular because it's directly built into GitHub. I don't know if the advanced GitHub, will you talk about GitHub Actions? Cool. The idea is that, when you're using GitHub, you have a piece of code on your machine that you push up into the GitHub repository, or the repository that lives on the GitHub website. And what GitHub Actions can do is basically take a set of tests that you've put into that repository and run them every time a new commit gets pushed into the repository, and tell you, did it work or not? And if it works, you get a little green check on, or a little badge you can put onto your site. There's a number of types of testing you can do. One is just to basically make sure that your code is of good quality. And so this is something that's really good to kind of work. If you're working in Python, especially, there's really good tools. For R, they're not quite as good. But basically using what we call static analysis tools, they basically look at your code and say, one, are there any obvious errors that I can see that they can see without actually running it, right? But also, are you adhering to the style guidelines, right? So for example, if I run this particular piece through PyLint, which is one of those tools, it can tell me, you're not supposed to mix tabs and spaces in Python, right? But you're trying to do that. you're redefining the list, which is a built-in variable, things like that, right? That you shouldn't do. If you code in, I guess you'll probably be working in Jupyter. I've become a big proponent of VS Code, which is the Microsoft development environment, which has really great integration of a lot of these tools, as well as other kind of code styling stuff that I'll talk about. But so, running, as I'll talk about in the NARP study, we had initially enlisted somebody to do a full code review of all of the code that I had written to analyze the results from the 70 teams. And they looked at my code and said, I'm not going to review it until you fix the style. And then I fixed it and I sent it back to them. They said, wow, it made it so much easier to read, once I fixed the style. Another thing that I think is important. Yeah, question. Oh, sorry, I have to walk up there. Go ahead. Why don't you just tell me and I'll. Can Pylint detect syntax errors like missing commas or parentheses? Yes, in general, it can't detect everything, but it can detect a lot of syntax errors. So it can often save you time by looking at, before you try to go run the thing, especially if the thing takes time to run, just look and see, are there any errors that I didn't catch? And I will often do that and see, oh, hey, I did something dumb up there. Let me fix that first. So, it's often common for people who come from a more engineering background to have this feeling that it's referred to often as the not invented here or NIH philosophy, that I need to build my own version of this thing, right? Nobody else's version is good enough for me to use. I need to build my own version of the DICOM to NIFTY converter or whatever it is, that you need to build. And the problem here is that if you're using homegrown code, right, there's a much higher likelihood that there are going to be errors in that code, as opposed to code that comes from groups that work together, that use good engineering practices. So for example, scikit-learn is a great example of this, right? It's a machine learning package for Python that has a ton of people working on it. I think adheres to very good software engineering practices in general. And if you need to do machine learning stuff, it's much better, I think, to find a piece of software like this that can do it than writing your own. Now, sometimes you can't find something that does what you want. And a good alternative there is you're often going to be able to find something that does 98% of what you want, and then you just need to make a little tweak, right? So you can take scikit-learn's code, it's open source, you can go change it yourself, and if you want to, you can even submit it back to them to be included in the package. That often takes a while, but for them to actually get to it. But you can do that, right? So I think that whenever you can use something that's well tested, that's a good thing to do. So are you okay with me going to noon, or did you want me to? Okay, cool. We will have a bit of time for additional questions in the end. I just want to kind of finish with a case study of the analyses that we did for the NARP study that I talked about earlier. So I've already told you about what we did. I'm going to talk about the workflow that I wrote, where we took all of those individual statistical maps and sort of processed them to give us overall results. And talk about some of the steps that we took. The first step was data versioning, right? So we want to know exactly, if I want to build the goal was to build a workflow that anybody could go reproduce from scratch. And the first thing that they need to know to do that is, what exact data set did you use for this, right? The data set changed over time. We had to fix problems with it. Data sets almost always change as you realize you need to add something or fix something. And so we basically created, first we start with the raw MR data, which are publicly available on OpenNeuro. They have a digital object identifier, which is a unique identifier that points you to a specific version of that data set, version 1.0.4. The data are shared according to this thing called the brain imaging data structure, or BIDS, which is basically a scheme for organizing both data, how do you name your files, how do you name your folders, what goes where, and the metadata, what data are required to be specified for a particular data set, how do you format those, and so on. there's, as I mentioned, there's a lot of work going on in the bids world. And this allows us to very easily take any data set and know immediately what to do with it. We also wanted to build a way for people to get as close to exactly the same answer as we got. And what we know from a lot of work is that, things like the version of software libraries can change the results that one gets. Often small, occasionally not so small effects. And we're inspired by this quote from my colleague David Donahoe. An article about a computational result is advertising, not scholarship. The actual scholarship is the full software environment, code, and data that produce the result. And so not a perfect solution, but a solution that we've sort of moved towards that I think solves a large number of the problems that we want to solve is the use of containers. And I'm sure you're going to hear more about containers through the week. The idea is that, this container has in it all of the software that you need and all the libraries that you need to run the software. So except for like weird numerical things or, random number generator things, you can get the same answer when you rerun the same code, even on a different computer. How many of you use R? Okay. Our world's a little behind. It's tricky to figure out how exactly do you, or I think it's gotten less tricky because there's other tools for this now. How do you make sure that I have exactly the same code versions? The problem is that if I say, go install ggplot, it's going to also install all the dependencies, but I could tell it to install a particular version of ggplot, but there wasn't really a good way to tell it to install particular versions of all of the things that ggplot is also going to install. So we started using this thing called checkpoint, which is pretty cool because it basically says, install all the versions of all my packages that were active in the R archive as of a particular date. And that worked pretty well for us. It takes time because it has to go like reinstall a bunch of stuff every time you first set it up. But nonetheless, we found it really useful. Another like small tool that I think is really useful for working into your workflow is the makefile. So a makefile is something that works on any Unix system, right? Works on the Mac. It allows you to basically specify a set of commands that you can very easily execute. So for example, instead of having to type in this long thing to run the NARPS analysis, I just say make run all, right? And you can also, for example, put in your style checker, say make check style, and it'll just go, check things for you. It's a really handy, and basically this is all put into a text file. And so it's a very handy thing to make it very easy for people to run complex workflows just from the Unix command line. Another thing we did was all of our analyses were implemented as software tests. And what that allowed us to do is then give those tests to one of these continuous integration systems. We use this thing called Circle CI. You would probably use GitHub Actions. And basically it ran all of the analyses as tests and stored out the data. And one, it would tell us, did it all work? Did it crash? And did things, I'll show you some of the kind of things we tried to confirm. But it also helped us make sure that we could make all that stuff run and have the outputs in a centralized place. We also implemented automatic code quality assessment. I don't know how important this is, but it was sort of useful just to see what it tells you about the potential problems in your code. So we use this thing called Codicy, which you just link to your GitHub repo, and every time you push new code, it'll basically give you a little report on code quality. I mentioned we did code review. So in this case, we Tristan Gletard, who's a skilled programmer, basically went through the entirety of the code, and we also had one other person dig through a bunch of it, and basically just looked it over to say, does it make sense what we're doing, right? Is it readable? Does our logic make sense? And also just help look for mistakes. And they found a number of things that we had done that were occasionally wrong, often suboptimal. So something that we've become also really interested in is, before we ever apply a piece of code for a real data analysis, try to run that same code on synthetic data where you know what the answer should be, and make sure that your code gives you back the answer, right? Because I think a big problem is, when we can put our data into our, we're kind of writing our analysis code on top of our data, and we get an answer, and we don't have, in biology, like, when you take your COVID test, right? It has a little control string, right? That's like a positive control, right? It's basically saying, if that doesn't show up, that's a signal that should be there. If it doesn't show up, then something's wrong, right? You'd also sometimes like a negative control, right? To make sure that if nothing's there, it will also tell you that nothing is there, right? So we've started trying to do that with projects in the lab when we can, especially for behavioral projects. It's easier. For imaging projects, it becomes a little more challenging because we don't have tools for generating good synthetic imaging data. But the idea is like one thing we like to do is parameter recovery. Like if I put in a known answer, can I get back that answer from my code? The other is kind of null testing. That's like our negative control. If I put in randomized data, will I get back results that tell me that there's no signal there except at my kind of error rate? And there's a number of good tools for generating synthetic data. This synthetic data vault is pretty cool. We've started playing with. For fMRI, the Brainiac toolbox has a fMRI simulator, still growing and pretty young, but I think it's there's a number of tools that are coming around. And so the one for the NARP study, what we did was we basically generated a simulated data set based on the known maps, and also included like the noise teams and the anti-correlated teams, and then had different levels of variance. So tried to basically generate random data or generate synthetic data that actually have signal in them based on the real data, and then compare the results of our final analysis to the ground truth that went into generating those synthetic data, and make sure that, for example, nothing's ever going to be perfect because there's noise in the system, right? But everything should correlate like above 0.95 between what we expect and what we actually get. And so we think that hopefully, there's pretty much no such thing as bug-free software, especially for a really substantial software project. This had thousands of lines of code. And so certainly it isn't bug-free, but we try to do everything we know how to do to improve the likelihood that if there's something wrong, we will find it. Okay, so I think I probably convinced you that we have a reproducibility problem. Statistical power is obviously one part of the puzzle. It's not the only one. And we don't want to let a sole focus on statistical power kind of blind us to other issues or other approaches that might be able to work with much smaller samples. But I hope I've convinced you that that there are tools out there coming from software engineering that can help us do a better job of writing code. Some of them take more work, some of them don't take much work at all to implement. And I'm certain that what you're going to learn here in the next 2 weeks is really going to help you do better science. So thank my lab, which still looks like this right now, and tons of awesome collaborators, some of which are in the room. Thank you. We have time for questions.
And predictions about how to work with
yeah. So right. So what do you do when you have a PI, you're a junior person and you're working with a PI or a senior person who doesn't want to share data? That's a really hard problem because obviously there are power dynamics that are at play. If you're a grad student, it's really, really difficult, right? I would say that one thing I have seen is I have known people who were several years ago resistant to data sharing, who are no longer resistant to data sharing, And the reason that they're no longer resistant to data sharing is primarily because their graduate students and postdocs made them come to realize that they have to engage in data sharing if they want to, remain part of the research culture and be, respected. And that's, those are, I mean, the hardest part of all this stuff is like, behavior change, right? It's like social and psychological stuff. It's not like technical stuff, right? Implementing the stuff I talked about here is easy. This is the hard stuff. It's like figuring out how do we get people to change their minds and change their behavior. And I think that one of the only ways to do that is just to keep pushing and to show as many examples as you can of other groups who are sharing their data, right? And there are some papers that claim, well, if you share your data, you get more citations. I think I don't know if you want to make too much of that, but I think it is certainly the case that if you're sharing your data, that your science comes to be viewed as at least more transparent. It's not necessarily better science in the sense of like getting the right answer, but it's certainly gonna certainly more transparent. It allows others to double check what you've done. So mostly I'm just commiserating that it's a really hard problem, but offering hope that that if you keep kind of pounding on the wall, that at some point it might start to move.
Balance on this kind of. Well,
That's a great question, right? So just to repeat the question, it's how does one kind of do this balancing act of like there's only so many hours in the day when I'm not like sleeping or hanging out with my wife or playing guitar or whatever else I want to do in my life, and I have to figure out how do I like find time to do this, like learning all these wacky tools and like learning about like new MRI methods, or like actually like thinking about scientific questions and what are the what does my lab want to go do next. And for me, it's I mean, one thing to say is I really like, kind of learning the technical stuff. And so I allow myself probably to spend more time than I should on that. I can now, right? I'm in a privileged position where, I have to do something pretty bad to be fired from my job. And so I'm really lucky that I can do that. And that's part of why I try to distill what I learned from that and, kind of hand that knowledge off to you and people in my group and others who I talk to. I think that and I think I'm opposite of most senior PIs, right? Most senior PIs spend increasingly less time writing code and doing hands-on stuff and more time like writing grants, but also like doing like bigger picture types of stuff. And I wouldn't have fun if I couldn't do this sort of stuff. And so that's why I make sure that I can find time to do it. So really it comes down to like, why are you in science, and what is it that gives you the most sort of pleasure about all the activities you have to do. And obviously we can't spend all of our time doing stuff that we really love to do. I have to go to committee meetings and write reports and all this kind of stuff. But figuring out like what are the things that you think are kind of close enough to your core set of skills that will like actually have some benefit for your science and for the people around you. But also just what do you have fun doing?
That are kind of stuff behind paywall, like something like like MATLAB or
Yep.
Brain Voyager, what would you tell them to do now?
Yep.
In order to better participate?
So the question is, what would I say to somebody who is stuck in a world where they're having to use non-open proprietary tools like MATLAB or Brain Voyager or whatever else it might be? My first thing I would say is like run like hell, right? I mean, because those are intrinsically not open, right? I can't, unless I'm going to pay however much I have to pay to go get a MATLAB license, or my university happens to have one, I can't reproduce what you've done, right? And that's going to mean a lot of people in the world can't reproduce what you've done. So I would certainly say, obviously, but we all have to make, we can't let the perfect be the enemy of the good, right? If it's the case that like the tools you really need only exist in MATLAB, then use MATLAB, right? And just try to do your best to write good MATLAB code and share your MATLAB code and test it as well as you can. I'm not a purist by any means, right? And I think that we ultimately, we're here to solve a problem, right? Of like figuring out, for most of us, like figuring out how the brain works. We think that using, open tools, being transparent, sharing data, all these sort of things will make it more likely that we get the right answer to those questions, right? But I wouldn't want to like put so much weight on the tools that you forget about the questions. And sometimes you just have to make a trade-off.
Slack about code review, asking a little bit more, how do you implement that in your labs, or what is the mechanics?
Yep. Yeah, so what we generally do is, and we've found this to be reasonably useful, somebody volunteers to have a piece of code reviewed, generally not very long, maybe, certainly no more than like a couple hundred lines of code. And it's often, a particular analysis that they're trying to code up or something like that. And so they send it out to everybody. And I've generally been the one running the review, which I would like to get away from, but that's just, how it's worked so far. So I'll put it up on the screen, and generally I'll ask them first to introduce just what's the problem you're trying to solve here, without giving me any details, just what's the overall problem this code's trying to solve. And let's say it's, well, I'm trying to like implement a drift diffusion model on this particular type of data, right? Then I read through the code line by line and basically try to see whether I can understand what the code is doing without them having to tell me anything. And often it'll be like, well, what is this variable? This variable is called this, but it's really, it's called like reaction time, but it's really something else. Like what do the variables mean? Are they named in ways that, a lot of it just comes down to like code formatting and variable naming and stuff like that, just to make sure that when I read the code, I'm actually getting what it's actually doing. And then I will often kind of refactor on the fly. So like I will, I'll have the code open. We've only done it with Python so far. So I'll have it open in my VS Code and I'll be, kind of like hacking on it as we go. And sometimes just put in comments, but sometimes actually, change something. And then I'll push that refactored bit back to them. And then they can, kind of take it and make those changes they actually want to sort of introduce them. Yeah, so the question is about sort of legacy code. Like every lab has code laying around that runs things that every new grad student or postdoc walks in and they're like, how do I pre-process my data? And I say, go run that script. Here's a description of how to run it, right? Don't ask what's going on inside because it's old and ugly. People have been using it here since like 1998 or whatever. And sometimes if you want to get stuff done, you just have to do that, right? And the question is like, how do you think about moving that towards open science practices? And I think, there's two there's two approaches, right? One is like sometimes, let's say it's a pre-processing workflow, right? Sometimes there is going to be another option that you can just plug into there, right? So for example, if you decide, well, we're going to move from using our old bash script to using fMRI prep, right? And there's work for doing that, right? Because the inputs might look different, the outputs are certainly going to look different, and you have to figure out how do you work with those. But ultimately, it might be the case that, you know, if something has been laying around the lab for 10 years, it's probably not a best practice kind of a tool, right? It probably has a lot of like outdated stuff in it. And, you know, I think it's generally good to be sort of, you know, changing code all the time to just update the tools that you're using. To make sure, you know, there's this concept called technical debt, right? Of like, you know, as code gets older, it relies on lots of things that start being outdated or ultimately break. And so code, you know, one of the really interesting things that comes out of the software engineering world is like the hard work of building software is not initially building the software. The hard work of building software is maintaining it and making it keep working over time. And that's really that's why simplicity and clarity are such important goals, right? Because other people are going to have to walk in and look at the code. And if they can't understand what's going on there, then that's going to make it really hard to make any kind of changes, right? So, so I feel for individuals who are in that sort of situation, but I've been there, right? And if you can't make a wholesale move to another platform, then another thing to think about is just like, are there small parts of it where you think that, you know, some improvement could be made, right? And start just making small changes. And the thing is, you need to make sure that you have a way to test to make sure that those changes are actually doing the right thing. And so that also becomes an opportunity to think, well, how do we actually validate this workflow, right? Presumably that 10-year-old code is not being validated. You don't have like a test suite for it or anything. You don't have data that you run through to make sure it gives you the right answer, or even that it just runs, period. So that's another thing to start thinking about before you ever, because one of the, there's this idea in software engineering of refactoring, right? Which is basically changing the software to make it better without changing what it actually does, right? It gives you the same output, but it just does it in a way that's generally more maintainable or more robust. And the first thing you need before you can do that is to have tests that tell you, did it do the thing it's supposed to do? So before you ever start thinking about writing the code, maybe start thinking about putting together a testing validation framework to just make sure it's doing the right thing. Let me get halfway up there and then I can hear you probably.
Some countries or institutions that prohibit open sharing fMRI data, even if they.
Yep.
What are the implications?
Yeah. So the question is, what do we do about the fact that there are certain jurisdictions or institutions that make it very difficult to easily share raw MRI data? And certainly, GDPR in Europe is, I don't know if that's what you're thinking of, but that is a framework that makes it much more challenging. In the US, we can, in theory, remove, all the identifiers like your name and all that stuff, and remove facial structures and maybe skull structures, and then share those data. Those data are then de-identified and actually treated as human subject data under US federal regulations. There are ongoing questions as to whether that's actually a good idea, right? There's some work suggesting that even data with the face removed can be re-identified with some level of accuracy. That hasn't risen to the level of concern for, we've talked about this with our legal people at Stanford, and they're not yet concerned that it's above a bar where we should stop sharing defaced MRI data. But I guess, my response to the question is there's almost always a way to share data. It really depends on what are the hoops you're going to have to jump through. How many lawyers are you going to have a conversation with? We now receive data from people in European institutions. It just requires paperwork and lawyers talking to lawyers. So it's generally doable. It's just a question of how important is it for you to share the data or to get your hands on the data in relationship to the amount of work it's going to take.